going to start by taking a look at the homework and try to get familiar with what we went over last week on chromosomes and mitosis and meiosis, or mitosis. Starting with the genome, or which of these definitions describes what a genome is?
The entire genetic complement of an organism.
Yeah, so that'd be the entire genetic complement of an organism, right? So the entirety of your DNA, that is going to make up your genome. Perfect. Now I'm going to skip homologous chromosomes for a quick second. I'm going to jump down to chromosomes. So what is a chromosome? Anybody got this one? So it's not a specific nucleotide.
A linear DNA molecule, yeah.
Right. It's a linear DNA molecule, right? So that DNA molecule is going to be bound to different proteins, okay, in order to kind of help keep it compact. Perfect. Okay, what about homologous chromosomes? What are homologous chromosomes here?
A set of chromosomes with the same length and the same genes.
Yes, absolutely. So it's a set of chromosomes that have the same length and the same genes in the same locations. So they're going to be almost identical chromosomes, but they're going to be coming from different parents here. Perfect. So they're going to be corresponding chromosomes. Cool. What does it mean to be diploid? What does it mean if a cell is diploid? So Kim, we said one set of chromosomes. So it's not one set of chromosomes, but this is where it's going to have two sets of chromosomes. Right. So if you're a diploid cell, you have two sets of chromosomes. Now, if you're a haploid cell, how many sets of chromosomes do you have?
One.
One. Right. So a haploid organism or a haploid cell would have one set of chromosomes. Perfect. Cool. Any questions on these definitions so far? All right, cool. Okay, so looks like we're good. Okay, so now with a gene, okay, so how would we describe a gene?
Specific nucleotide segments on DNA.
Right, it would be the specific nucleotide segments on your DNA. Right, so remember, genes are specific regions on the DNA that are going to code for some trait. Okay, so everyone have the same genes, but the variations within those genes, the variations of those nucleotides, is going to create the variation that we see in various populations. Perfect. Okay, what about a locus? What is a locus? A location on a chromosome, right? Perfect, Michaela. Yeah, so a locus would be just a specific location on the chromosome. Perfect. What's the cell cycle?
Series of events involving cell growth and division.
Yeah, so it's going to be a series of events involving cell growth and division, right? So for the first portion of the cell cycle, the cell is going to be just growing. And then for the last sliver of it, it's going to be actually going through division. So the whole cycle of growing and division is going to be called the cell cycle. Beautiful. Okay. What is a centrosome? So centrosomes are not the microtubule apparatus itself, but what is it going to be?
The organelles that create mitotic spindles.
Right. They're the organelles that create the mitotic spindles. Perfect. So the organelles that create the mitotic spindles are going to be the centrosomes. So they're going to get produced or duplicated during interphase, and then they're going to help you start to produce those microtubules, those mitotic spindles. Perfect. And the last one is the mitotic spindles. Which one would this one be? That would be exactly, so Jackie got it right. So the microtubule apparatus, and so these microtubules are going to make up the mitotic spindles. And so those mitotic spindles will then eventually bind onto the chromosomes and then move them around during mitosis. Perfect. Okay, any questions on any of these definitions so far? All right, looks like we're good. All right, so starting with question 2. So an organism's traits are determined by the specific combination of inherited genes, cells, proteins, or chromatids. Yes, he says genes, and that is absolutely right. So, and Kimberly's got that one good too. So yeah, so organisms' traits are going to be determined by their inherited genes. So remember, the gene is just a specific region on your DNA, but the different nucleotides within that gene is going to contribute to the different variations within our population. All right. Question 3. So at which stage does the cell become committed to cell division? So the G1 checkpoint, the G2 checkpoint, M phase, or prophase? Yes, beautiful. Everyone's getting this one right. So this is the G1 checkpoint. Yeah, so once you go past the G1 checkpoint, okay, that's the point of no return. There's no going back. Once you pass the G1 checkpoint, you are committing, or that cell is going to be committed to undergoing cell division. Perfect. Okay, question 4. Which of the following does not take place during prophase? So we got the chromosomes are lined up at the metaphase plate. The nuclear envelope breaks down. The spindle fibers begin to emerge from the centrosome, and then chromosomes begin to condense. Right, so everyone's getting this one right. So chromosomes are not going to line up at the metaphase plate. Okay. What phase do they line up at the metaphase plate at? Metaphase, right. Yeah, so they're going to line up at the metaphase plate during metaphase. Okay. Beautiful, pretty straightforward. Okay, question 5. Which of the following is the longest phase of the cell cycle? Okay, so we got interphase, mitosis, metaphase, or G2 checkpoint. Interphase, yes, everyone's getting this one good. Perfect. So interphase is the longest phase of the cell cycle. So the cells are going to spend the majority of their time in interphase. So if you remember going back to lab last week when you guys were looking at those cells under the microscope, okay, the majority of the cells that you should have seen should have been in interphase because that's where cells are going to spend the majority of their time in. Right. Beautiful. Okay, so question 6. Which of the following comes after telophase? So we have cytokinesis, prophase, interphase, or anaphase. Cytokinesis, right. So someone tell me what is cytokinesis? Okay, what happens during cytokinesis? Right, so yes, you got it. So during cytokinesis, you're gonna have your cells actually begin to split into 2 individual independent daughter cells. Okay? In telophase, you still have 2 nuclei, but they're all confined to a single cell. Okay? So once cytokinesis happens, they pinch off and they separate, and now you end up forming 2 new daughter cells. Perfect. Okay? And that's gonna happen during cytokinesis. Awesome, cool. Any questions so far in any of these? Okay, cool. All right, so now next question. So we have question 7. So what is the importance of the M checkpoint? Okay, so we got A, it ensures the spindle fibers are correctly attached to the kinetochores. B, it ensures that the cell has reached a sufficient size. C, it ensures that an adequate stockpile of nucleotides has been obtained. Or D, ensures accurate and complete DNA replication. Yes, so this is correct. So everyone's got this one right. So it's going to ensure that all the spindle fibers are correctly attached to the kinetochores. Okay, so on Thursday, we're going to see what's going to happen if those kinetochores are not attached to spindle fibers properly. So stay tuned for that. That's going to be really interesting on Thursday. Perfect. Okay. Question 8. So how does cytokinesis occur in animal cells? Okay. So A, we get an actin ring forms around the metaphase plate and constricts the cell until it splits in 2. B, a cell plate develops between the 2 nuclei, separating the cell into 2. Or C, an actin ring and a cell plate develop between the nuclei. Right, so everyone's got this one. So the actin ring is going to form around the metaphase plate. And when it does that, it's going to start to pinch and pinch and pinch and pinch until that cell eventually splits into 2 individual daughter cells. Perfect. Okay. How does cytokinesis happen in plant cells? Okay. If we're looking at a plant cell undergoing cytokinesis, what's that going to look like? Right, you're going to have that cell wall grow in between them. Right, so you're going to have a cell plate develop, and that cell plate is going to grow until it eventually becomes a new cell wall. Beautiful. Okay. Question 9. So a particular inhibitor is used to temporarily pause mitosis. Okay. After using the inhibitor, we observe the cells through a microscope and see that all the cells are in the stage shown below. Okay, this is what our cells are looking like. Okay. So what stage of mitosis are these cells paused at? Anaphase. Perfect. Yes, beautiful, beautiful. So they'd be paused at anaphase. Wonderful. Good. Question 10. So which of the following correctly describes the roles of S phase? Okay, so we have A, the chromosomes are replicated, B, the ribosomes and proteins are produced, C, the cell continues to grow, or D, the final preparations for entering the mitotic phase are made. What happens during S phase? Yes, so a bunch of people are getting this one right. So yeah, so chromosomes are going to be replicated during S phase. Wonderful. Okay, and then last question, question 11. So which stage of mitosis is shown below? This is metaphase, anaphase, prophase, or telophase. This is metaphase. Beautiful. And so we can tell because all of our cells are lined up in a single file line, okay, along the metaphase plate. Beautiful. Okay, any questions on anything we've covered from the homework or anything from the last lecture? Okay. All righty. So if there's no questions, we will go ahead and jump into the homework for today, or the PowerPoint for today, or like today's lecture. And so, is everyone able to see the PowerPoint? Cool. All righty. So today's lecture, we're gonna kind of be talking about 3 separate topics that don't really have much to do with each other. So we're gonna be kind of, today's lecture is gonna be a little bit choppy and not really gonna flow. But we're just kind of got to get through it because there's stuff that we're going to be focusing on Thursday that's going to be helpful for this this material here. So just kind of bear with me. It's going to be choppy. There's not going to be any kind of flow. We're just going to hit 3 different topics all today, but they're all going to be relatively short. Okay, so bear with me today. Thursday, everything is going to flow just like normal. Okay, so today our first topic that we're going to be covering is cancer, okay, and how cancer comes from the cell cycle. Okay. So we've probably seen these signs around, whether we go to Disneyland or whether we go to, different restaurants or different establishments, where you're going to see a sign that says that there's some chemical or some material that's used at that establishment that may be known to cause cancer. Has anyone seen these signs before? Okay. Yeah. So these are pretty common signs, and these, like Mario said, these are all over California. And so this is important because a lot of these different chemicals can cause cancer. Now, not all of them will cause cancer, but a lot of them have been known to, be contributors to developing cancer. Okay? And so when we look at cancer, we're going to be looking at how cancer is actually going to develop. Okay? So we have a quick little diagram over here. And so what happens during to develop cancers, we have some kind of normal cell that gets exposed to a cancer-causing agent. Okay? When that gets exposed, it causes some changes within the cell, and that eventually will develop into a cancer cell. Okay? And so today we're going to be talking about how cancer cells can develop. Okay? So we haven't talked about this already, but just kind of gauge where your minds are at, because it did kind of show up in lab a few weeks ago. But very, very, very briefly. Okay, so does anyone know, how does cancer develop? So we got A, the cell cycle becomes unregulated and occurs too quickly or too often. B, chromosomes in metaphase do not line up properly along the metaphase plate. Or C, mutations in DNA arise. What do we think? So I'm seeing a combination of A's and C's. Okay.
A.
The main answer is A. So the cell cycle becomes unregulated. Okay. And so because it starts to occur too often, and so because the cell cycle starts to occur too often, you are not able to catch mutations in the DNA. Okay. And that can lead to cancer. Okay, so it's a combination of A and C, but the main thing that causes cancer is the cell cycle becomes unregulated and starts happening too fast. Okay, so that's that. Next question. So what are the main roles of the G1 and the G2 checkpoints? Okay, so we got A, they ensure the sister chromatids are attached to chromosomes correctly, or B, they check for damage in genomic DNA. What do we think for this one? What do we think? Yes, I'm seeing some B's. Yeah, so B is the correct answer. So there's going to be an error, or they're going to check for damage within the DNA. Okay. So those are the roles of those two checkpoints. So we're going to be taking a look at how we can end up developing mutations during the process of the cell cycle happening too fast, which will ultimately lead into cancers. So here's how this whole process is going to work. Okay. So cancer is going to be caused by uncontrolled cell division. Okay. Cell division should be happening at a specific rate. Okay. There should be a specific rate to how the cell cycle should be working. However, if that cell cycle begins to go uncontrollably and starts going too fast, okay, we can end up developing lots of cells that are non-functional and developing these big balls of cells that are non-functional cells here. Okay. And those very large pieces of cells, those very large clumps of non-functional cells that develop this way are what we call tumors. Okay. So the way this works is that during DNA replication, sometimes there's some errors in DNA replication. Okay. So here, for example, we're supposed to have a C and a G, but let's just say when we're replicating the DNA, okay, we end up with an issue or an error, and we end up forming a T and an A at that particular point. Okay. So we have an error. Okay. At that point, the cell needs to recognize that, hey, there's a problem here. There's an error that we need to fix. Okay. And so in most cases, the cell will say, hey, oh, hold on, there's a T and an A here. Let's switch this back because this isn't right. This needs to be a C and a G. Okay. And then switch it back, everything is good. Okay, you're good to go. Okay. However, there are some issues or some scenarios where this error doesn't get recognized. Okay, it doesn't get caught. And so you end up with having this error kind of continue to exist inside the DNA. Okay. And so that is what's called a mutation. Okay. So a mutation is where you have a region of your DNA, okay, that is not correct. Okay. So here we're supposed to have a C and a G. Here we have a T and an A. And so this would be considered a mutation. Okay. So mutations can develop, okay, even when the cell cycle is controlled properly. Okay. The difference, though, is whether that error gets caught. Okay. If the error gets caught, the mutation gets fixed, everything is good to go. If that mutation doesn't get caught, then the cell can then continue to replicate and pass on that mutation to the future generations of cells, okay, to the next daughter cells. Okay. And so when you have a mutation in your DNA that gets passed on to the daughter cells, okay, it now becomes easier to develop further mutations. Okay. And so now if you keep going through this whole process over and over again, now you're going to have a bunch of cells, okay, that have mutated DNA, and so they're not going to end up working properly. Okay. And so you have these big balls of cells that don't work efficiently or don't work like they're supposed to, and you end up getting tumors. Okay. So everyone kind of following me so far? Okay. If I'm making sense, go ahead and throw a checkpoint or a check mark or a star up here. Okay. Yeah, cool. Okay, so cool so far. Okay, so I'm seeing some stars. Okay, so cool, glad. Okay, all right, so the way this develops is through the DNA, okay, as we know. So DNA, like we talked about on Thursday, okay, DNA has a role in helping produce proteins, okay? And those proteins are gonna give you the particular traits you're looking for, whether you're looking at hair color, hair texture, if you're looking at eye color, if you're looking at temperament, if you're looking at development of certain structures, okay? DNA is gonna help to produce proteins, okay? So the specific parts of the DNA that are going to be producing those proteins are going to be specific genes. Okay? So genes are essentially going to be, they're essentially going to be the instruction manual okay, to help you produce your proteins. Okay? To answer Hasmin's questions, if you go to annotate, there should be a button called annotate, either at the top of your screen or at the bottom. If you hit annotate, you go to stamp and it comes up with a star and so on and so forth. Okay, so you end up with genes that are going to be producing DNA. Okay, so the genes are the instruction manual. So if you can kind of think about, if you want to make Hot Pockets, for example, right? There's going to be instruction manual on how to produce those Hot Pockets. Okay? Now, if there's a mutation in that DNA, okay, that means we have an error in the DNA. We have an error in our instruction manual. Okay? And so if we have an error, okay, we're not going to be able to build that protein properly. Okay? That protein that we produce is either going to be less functional or non-functional. Okay? Same thing as if we're trying to build our Hot Pockets, right? If there's an error in our instruction manual, let's say our instruction manual, forgets to tell us to put cheese, right? We're going to end up with Hot Pockets that don't have any cheese, right? We don't have a functional product. We don't have a functional Hot Pocket. Same thing is going to be the same as when we have our DNA. Okay, if our DNA gets mutated, okay, the protein that we end up producing is not going to be functional, or is not going to be as functional as it should be. Okay, and so that creates issues when you're starting to produce different things throughout the cell or throughout the organism. Okay. Okay, so that's how mutated DNA can kind of play a role into developing proteins, okay, or non-functional proteins. Okay. And so when you start to develop all of these different mutations, the cell cycle begins to speed up, okay, because you start pushing past a lot of those checkpoints, the G1 checkpoint, the G2 checkpoint, and the M checkpoint, right? If you start pushing past all of those checkpoints, then the repair mechanisms at those checkpoints become less effective. Okay? So the checkpoints themselves become less effective because you're pushing past them before you reach those conditions, before you meet all of those conditions at those checkpoints. Okay? And then the repair mechanisms in those checkpoints become less effective because, again, you're not spending enough time in those checkpoints, and you're not giving the repair mechanisms a chance to actually repair the problem. Okay? And so you end up developing more mutations because you have no way of fixing it. Okay? And so what happens here is these mutations these mutated cells, I should say, these mutated cells begin to grow a lot faster than they should. Okay? And so they're going to end up growing a lot faster than the normal cells would. And so because you have a large number of mutated cells growing, okay, and these mutated cells not working as they should, okay, those clumps of mutated cells become the actual tumor. Okay? So for example, if you're looking at a bone cell, right, bone cells are supposed to allow bones to become hard. They're supposed to be strong, rigid, so on and so forth. Okay? But if there's a mutation in that bone cell, let's say that mutation causes that bone cell to become more brittle. Okay? It's not going to be as strong. Okay? That mutation is going to cause that bone cell to start pushing past the cell cycle a lot faster and pushing past all of those checkpoints, okay, before the conditions of those checkpoints are met. Okay? And so if that happens, okay, the cell cycle is going to happen a lot faster, and that bone cell is going to continue to divide and divide and divide and divide and start producing a bunch of bone cells that continue to have that same mutation, and maybe even more mutations as well. Okay? And so because you have a lot of bone cells that are starting to grow a lot faster than your normal bone cells do, okay, that clump of bone cells that is mutated becomes the actual bone tumor. Okay? Does this make sense? Am I kind of making sense here?
Yes, it makes sense to me.
Cool. All right, cool. Any questions on this process before we go forward here? Okay. Okay, cool. Okay. So there are specific genes that are going to be responsible for contributing to developing cancer. Okay. So cancer is going to develop when one of two types of genes are mutated. Okay? So the first type of gene is called a proto-oncogene. Okay? So a proto-oncogene is a normal gene, okay, in the DNA that encourages the cell to go through the cell cycle properly. Okay? And so it's going to cause the cell to just go through the cell cycle as it should. Okay? So proto-oncogenes are good cells, or good genes. Okay? However, if that proto-oncogene gets mutated, then it becomes an oncogene. And that oncogene is going to start resulting in that cell becoming cancerous. Okay? So here's the way this works. Okay? So typically, if there's a mutated DNA sequence, okay, the DNA sequence is going to cause the production of either a less functional protein or a non-functional protein altogether. Okay? If that's the case, okay, that can actually be detrimental to the cell, and that causes the cell from not being able to replicate. Okay? The cell is not going to be able to replicate if that DNA is mutated. Okay? If that cell is not able to replicate, it eventually will die. Okay, and then that mutation is not passed on to the next generation. That's a good thing. Okay, that's what we want to have happen if we have a mutation. Okay, because we don't want that mutation to continue to propagate into future generations. Okay? So that's typically what will happen if there's a mutated DNA sequence. Okay? However, occasionally what will happen is a mutated oncogene, okay, can cause the cell to continue to live. Okay? And so if it continues to live and still be able to replicate, it's going to push past all of those checkpoints. And so you start moving through the cell cycle a lot faster than you should and not stopping at those checkpoints. Okay? And so as a result, you end up passing that mutated gene, that oncogene, into the future daughter cells, because again, you're pushing past those checkpoints and you start going through mitosis before you should. Okay. And so as a result, all of the resulting cells, all the resulting daughter cells that came from that mutated parent cell will all have that oncogene, okay, and they'll all continue to push past the cell cycle and push past that the checkpoints before they should. Okay. And they start replicating too fast and you end up developing cancer. Okay. Okay. So that's how proto-oncogenes will work. Okay. So an example of a proto-oncogene is called cyclin-dependent kinase, or CDK. Okay. So there's a CDK gene, okay, in the DNA, and that's going to code for a CDK protein. Okay. And that protein, the CDK protein, cyclin-dependent kinase, is going to be involved with regulating your cell cycle. Okay. So this is the type of proto-oncogene. Okay. So if CDK gets mutated, it's going to cause the cell to push past the cell cycle before, or push past the cell cycle checkpoints, okay, before the conditions of each checkpoint is met. Okay, so it's going to push past the G1 checkpoint before those conditions are met. It's going to push past the G2 checkpoint before those conditions are met. Okay? And it's the same thing with the M checkpoint. Okay, and you're going to start going through mitosis before you have checkpoints that are that have conditions that are met. Okay? Now, one of two things can happen at this point. Okay, scenario one, okay, is the cell cycles, or sorry, the daughter cell's DNA is too damaged. Okay? If the daughter cell's DNA is too damaged, okay, then it's not going to go through the cell cycle anymore, and so you won't produce any more mutations. Okay? Again, that's a good thing. That's what you want to have happen, because you don't want to start developing more mutations, okay, or more cells with that same mutation. Okay? So if we destroy that daughter cell ends up dying and not going through cell division, okay, that's a good thing. Okay? Scenario two is if that DNA is still salvageable, okay, if that mutated DNA is still salvageable within that daughter cell, okay, that daughter cell can then continue to go through the cell cycle and again push past all of those checkpoints before those conditions are met. Okay. And so as a result, the daughter cells that result end up having more mutations, okay, and so they can develop more mutations and so on and so forth as they continue to divide over and over and over again. Okay, and so you can end up having daughter cells that produce even more mutations that you initially had, all because you're pushing past the cell cycle before you should. Okay, okay. Does this make sense? Okay. If it makes sense, go ahead and throw a star, go ahead and throw a checkpoint, or say something in the chat so I know I'm making sense here. Any questions? No? Okay, so if there's no questions, we'll go ahead and move on to the next type.
I have a question.
So then, oh yeah.
Do we all get cancer at some point then? I mean, I don't know. Just.
Yeah, so it depends. From what I heard from one of my professors in grad school, he was saying that pretty much everyone will develop cancer at some point. Usually it's going to be happening like way later on in life when you're kind of at your older ages. And so there's just certain scenarios where your body doesn't go through things efficiently, because your cells essentially don't work as well as they used to. And so the cell cycle, just like anything else in your body or anything else in life, okay, doesn't work as well as it once did. And so as you get older, sometimes those proteins that are involved in regulating your cell cycle don't work as well. Okay? And so you start to undergo or start to develop cancer because you just are not able to catch those mutations when they happen. Okay. Does that make sense?
Yeah, that makes sense. Yeah. Okay.
Yeah. Cool. Yeah, very good question. Very good question. Any other questions here? Cool. Okay. So those are the proto-oncogenes. Okay. Now there's a second type of gene, okay, that's going to be important, and that can also help to cause cancer as well. Okay. And so, or when they're mutated, I should say. Okay. So these types of these types of genes are called tumor suppressor genes. Okay. So tumor suppressor genes are similar to proto-oncogenes, that these are normal genes. Okay. So tumor suppressor genes are involved with stopping the cell cycle or halting it, okay, until those conditions are met during those checkpoints. Okay. So when you go to the G1 checkpoint, okay, the tumor suppressor genes are involved with stopping you at the G1 checkpoint until all those conditions are met. Then you move to the next one. Okay. Then the tumor suppressor genes will cause you to stop at the G2 checkpoint, okay, until those conditions are met. Okay. Then you'll go through, you'll stop at the M checkpoint. Okay. The tumor suppressor genes will stop you at the M checkpoint until those conditions are met. Okay. So they're in job of stopping the cell cycle at each checkpoint so the cell can make sure that all the conditions of that checkpoint are met. So these are normal genes. Okay. So there's three main types of tumor suppressor genes. There's the RB1 gene, there's the P53, and then the P21 gene. Okay. And they all kind of work essentially the same way. Okay. Now, if these tumor suppressor genes get mutated, okay, if they get mutated, they're not going to be able to stop the cell cycle at each checkpoint. Okay. And you start to run through all of your checkpoints again before those conditions are met. Okay. Especially if there's a problem in that DNA. So one specific type of tumor suppressor gene is the P53 gene. Okay? And so P53 is a tumor suppressor gene that's going to produce the P53 protein. Okay? And so P53 is a type of tumor suppressor gene that's actually, or when it gets mutated, it actually results in cancer cells. Okay? And so about over 50% of cancer cells result because of a mutation in the P53 gene. Okay? And so what it's going to do is it's essentially going to stop the cell cycle at the G1 checkpoint. Okay? And so what it's going to do is it's going to kind of do a couple things here. Okay? So when you have a normal P53 gene, okay, it's going to do one of two things. Okay? It's either going to cause the cell cycle to stop, okay, arrest the cell cycle here, okay, cause the cell cycle to halt. Okay? And so when it halts, it now has one of two options. Okay? Option A is it can stop and then allow some DNA repair. Okay. Once that DNA gets repaired, it will then cause the cell cycle to then restart. Okay. And so you go through the cell cycle all over again. Okay. Or you continue going through the cell cycle. Option B is if the DNA is too damaged, okay, if the DNA damage is too severe, it'll activate genes that cause a process called apoptosis. Okay. And so apoptosis is when the cell actually kills itself. And again, it's going to do that if the DNA is too damaged. Again, that's a good thing that we want to have happen because you don't want a cell that is mutated to continue developing new daughter cells that have that same mutation and possibly developing more mutations. Okay? So you want to kill that cell before it has a chance to do that. Okay? So those are the two options here. Okay? So P53, you have either the cell cycle arresting here. Okay? When it arrests or when it halts, it allows time for the DNA to get repaired. Once the DNA gets repaired, it will then kind of continue running through the rest of the cell cycle. Okay. Option B is if the DNA damage is too severe, it'll cause the cell to basically kill itself, going through apoptosis. Okay. That's how P53 should work, okay, if it's not mutated. Okay. Now, if P53 does get mutated, okay, it will not have the ability to determine if there's any damage in DNA. So the cell cycle is going to continue even if there's problems, even if there's mutations in the DNA. Okay. And so, for example, here we have our mutated P53. Okay. If there's DNA damage, it's not going to be able to recognize the fact that there is DNA damage, and it's going to continue to run through the cell cycle as if there's nothing wrong. Okay. When there is, in fact, something wrong with the DNA. Okay. Okay. So the cell cycle continues, and you start to produce cells that have a mutation. Okay. And that cell that has a mutation is going to continue to go through the cell cycle and divide and produce more cells with that same mutation and developing even more mutations going forward. Okay. So it's not going to be able to trigger apoptosis. It's not going to be able to cause itself to kill itself because it has no way of recognizing that there is a mutation in the first place. Okay. And so as a result, you end up developing cells that continue to have the same mutation, end up having a giant ball of cells that is all mutated and not working properly, and you end up with a tumor. Okay? And so that's how you end up with cancer as a result of the P53 gene. Okay. Any questions on tumor suppressor genes? So this kind of makes sense. No? Okay. Cool. Okay, so any questions here? Okay. So if it kind of helps, so Amayali's question, so is there no way to stop it? Right, so if the P53 gene is mutated, okay, then there is essentially no way to stop it. There's a few other points in the cell cycle that can be where you can check it, okay, but usually if P53 doesn't catch it, okay, then it's usually just gonna kind of, there's pretty much no way to stop it at that point. Okay, it's just gonna kind of continue, especially because it's gonna have its role in the G1 checkpoint. So at that point, if it passes the G1 checkpoint without recognizing that there's a problem, it's just going to continue to go through as if there's no problem. Okay? So there's really there's really not much of a way to stop it at that point. Yeah. So if it kind of helps, I was kind of running through this whole scenario with my wife, and so she she's kind of a her kind of drinking choice is tequila, for example. Okay, actually, let me get Mara's question. So Mara's question, so is there a way to check if your tumor suppressors have mutated or changed them? There really is not much of a way to check. There are specific sciences that you can do, or specific kind of processes that you can do to kind of look to see if that particular gene is mutated, but it's kind of difficult to do that. And so it's possible, it's just a very extensive and kind of difficult process and a very long process. So one way that you can kind of think about the P53 gene is if you're kind of looking at like someone who's drunk, for example, right? So if there's someone who's drunk, the way a normal P53 gene should work is, let's say you got a drunk person who's supposed to be driving home, or is not supposed to be driving home. Okay, they know they're not supposed to be driving home, and they end up driving home. Okay, they end up getting arrested. Okay, so they get arrested by the cop. Okay. Once the cop arrests them, they throw them in jail. Okay, let them kind of sit there. Okay, sober up. Once they sober up, they let them go home. Okay, cell cycle restarts. Okay. Okay. If there's a situation where this gets really bad, then they'll go through apoptosis and they'll just kind of be thrown in the drunk tank, and then they're kind of their own thing. Okay. But essentially, what's going to be happening here, again, if you have a normal P53 gene working, okay, the cell cycle is going to arrest. You're going to have some DNA repair, and then it's going to restart the cell cycle. Okay. If not, it's going to undergo apoptosis. Okay. All right, so that is the cancer. Okay, so next kind of topic here is going to be looking at prokaryotic cell division. Okay, so prokaryotic cell division is going to be happening very similarly to mitosis. So we're kind of shifting gears very, very intensely here. Actually, before we go any further, are there any other questions on cancer before we go forward? Any other questions? Oh, yeah, no problem, Kimberly. Okay. All right, so now we're going to look at prokaryotes and how they're going to undergo cell division. Okay. So their process of undergoing cell division is going to be a little bit different, okay, from mitosis. It's going to look very similar, but there's going to be some differences here. Okay. Before we get there, so what's the purpose of cell division? Okay. A, to help cells grow. B, to help cells produce new cells that perform different functions. Or C, produce daughter cells that are genetically identical to the parent cell. So I'm seeing C's. And C is correct, yeah. So the purpose of cell division is to produce genetically identical daughter cells. Okay, we want to have the daughter cells be genetically identical to the parent. Perfect. So next question. So how does prokaryotic cell DNA appear? Is it in a square formation, in linear formation, in a triangular formation, or in a circular formation? So I'm seeing these. Yeah, so D is correct. So it's going to happen in a circular formation. So prokaryotic DNA is going to be circular, whereas eukaryotic DNA is going to be more linear. Perfect. Okay, last one. So where does prokaryotic cell DNA reside? In the mitochondria, in the nucleus, in the nucleoid, or in the pili? What do we think? So seeing a couple of right answers here. So the answer here is the nucleoid. So remember, when we're looking at prokaryotes, prokaryotes do not have a nucleus, okay? So their DNA is going to be found inside the nucleoid, okay, with that region kind of in the middle of the cell. Cool. Okay, so prokaryotes are going to divide in a process that's going to be different from mitosis, but kind of look a little bit similar here. Okay, this process is going to be called binary fission. Okay, and so most eukaryotes, like bacteria and things like that, are going to be replicating through binary fission in order to produce daughter cells that are genetically identical to the parent cell. Okay, so again, you're just having a single cell splitting into two individual daughter cells, and then they're going to look exactly the same as the parent. Okay. And so prokaryotes, like bacteria, certain protists and things like that, they're going to be replicating through binary fission. Okay. Now this process is going to be different from mitosis because prokaryotes do not have a nucleus, okay, and because they only have a single chromosome. Okay. So for that reason, the prokaryotes are not going to be going through mitosis. There's not going to be a nucleus to break down, and there's not going to be, you know, a bunch of chromosomes to line up around here. Okay. You just have a single nucleus, or sorry, a nucleoid, and then a single chromosome. Okay. So that's kind of what we see here. We have this parent cell splitting into eventually form a daughter cell through this process. Okay. So the way this works is binary fission is going to, like I said, it's going to look similar to mitosis, but a little bit different here. Okay. The key with binary fission, though, is it's a lot less complicated than mitosis, and it happens a lot faster. Okay. And so one example where we can kind of see binary fission kind of happening is if we were to take our hand and put it onto a petri dish. Okay? So if you look at your hand's going to have a bunch of bacteria on it that you're not able to see. Okay? And if we put it onto this petri dish, okay, and you let that petri dish just incubate for a few hours or a day or 2 days, okay, you take that petri dish out, you're going to be able to see all of these different bacterial colonies, okay, all over your petri dish. Okay? And so each one of these little circles is a bacterial colony, okay, and in each one of these colonies, you're going to have anywhere from hundreds to thousands and possibly even millions of bacterial cells that all developed over a very short period of time. Okay, and the reason for that is because binary fission happens very quickly. Okay, it's able to replicate that cell very, very fast. Okay, so this is one of the reasons why it's important to wash your hands, especially now that we're dealing with this whole coronavirus situation. So, wash your hands here. Okay, anywho. So that's kind of how binary fission will be working here. So again, that chromosome that we're going to be taking a look at inside these prokaryotes is going to be found in a circular formation within the nucleoid. Okay. Now, kind of similar to eukaryotic DNA, okay, prokaryotic DNA is going to be wound around a bunch of proteins to help keep that DNA molecule compact. Okay. Compact and pretty condensed. Okay, so it kind of, and that's going to be important because when we look at DNA, if you were to kind of stretch it all the way out, can range for multiple miles, and so it needs to be compacted into a very small individual cell. Okay, so those proteins help to kind of compact the DNA here. Okay, so here's the way binary fission will work. Okay, so you're going to have your circular DNA, okay, the circular prokaryotic DNA, so in our bacteria here, and there's going to be a point on that DNA called the origin. At the origin, the cell is going to start replicating. So you're going to start replicating at the origin, and you're going to start causing that replication to happen in both directions away from the origin. Okay? And so that's what we see over here. Okay, so here we have our origin, and the replication is going to happen in both directions on opposite sides of the origin here. Okay? The origin is going to be located on the chromosome near where the chromosome binds to the plasma membrane. Okay? And when you start forming that new strand of DNA, okay, the new strand is going to start to move away from the cell wall attachment. Okay? And it's going to start moving to the opposite side of the cell. So that's the replication portion here. Once we replicate that DNA, okay, the cell is going to begin to elongate. Okay? And so as it elongates, okay, the plasma membrane is going to begin to grow, and that's going to help to move the chromosomes to opposite sides of the cell. Okay? And so as that chromosome starts to move to opposite sides, okay, we're going to start pinching at the midpoint here. Okay? And that's going to begin our process of cytoplasmic separation. Okay? This is where we're going to start to begin the cytokinesis process here, okay? Or a process similar to cytokinesis, I should say. Okay. All right. Okay. So what's going to happen here when we start to form our kind of our septum here, and the septum is going to be the point where this cell begins to pinch, okay? In between the nucleoids. Okay. So it's going to pinch in between the nucleoids here. And so what's going to happen here is we're going to form a process that's going to be similar to the cleavage furrow. Okay. So we have this protein called FITZ. Okay. And so FITZ is going to be a protein that's going to form a ring around the midpoint of the cell, okay, in between the two nucleoids. Okay. And so what's going to happen is FITZ is going to constrict and constrict and constrict and constrict and constrict until you eventually split that cell into two individual daughter cells. And then once you form your new cell walls, okay, the daughter cells will then completely separate at this point. Okay. So kind of a pretty short process compared to mitosis. Let's kind of run through this one more time. Okay. We have our origin. Our origin is going to be where we start to begin our DNA replication, and the DNA replication is going to happen in both directions away from the origin, okay, because it's a circular chromosome. As it starts to move away, the cell is going to begin to elongate, and that's going to help to move the chromosomes to opposite sides of the cell. Okay. And then we start to form a little cleavage furrow, okay, this little pinching point in between the two nucleoids. Okay. So we're going to form this protein called FITZ, and so FITZ is going to line up along the cleavage furrow, okay, and it's going to pinch and pinch and pinch and pinch until you eventually separate that cell into two individual daughter cells. Okay. And that's really all there is to binary fission. Okay. So because this is a very short process, these bacteria cells or other prokaryotes are able to replicate so quickly, which is why you're able to get these very large bacterial colonies over a very short period of time. Okay. Cool. Any questions on binary fission? Does it all make sense? If it makes sense, go ahead and say something in the chat, or give me a star or a check or something. Any questions here? Cool. All right, beautiful. Okay, so if there's no question, okay, so I'm seeing a good amount of stars and checks. Perfect. Okay, so next question here. So the action of FITZ in prokaryotes is similar to the action of what element of the cytoskeleton in eukaryotes during mitosis? Okay. So what does FITZ act similar to when we're looking at mitosis? Is it going to be the microtubules, the intermediate filaments, or actin filaments? What do we think here? So Kimberly says C. Anyone agree with C or disagree with C? Actin, yes. So that's exactly it. The answer is actin. All right, so the actin filaments are going to help form the cleavage furrow. So good job, guys. So the actin filaments are going to help to form the cleavage furrow in eukaryotes during mitosis. Okay? And so it's going to pinch and then cause the animal cells to split into two components. FITZ acts essentially the same way in terms of forming the cleavage furrow and pinching to separate that prokaryotic cell into two components here. So exactly right, beautiful. Good job. All right. So any questions on prokaryotic cell division before we jump into our next topic here? Okay. So if there's no questions, we'll go ahead and jump into the last one. And so this last one, we're kind of moving away from mitosis at this point, and we're going to be looking at a different type of reproduction. Okay. So over the last two lectures, on Thursday and on today, we were looking at basically asexual reproduction, okay, allowing prokaryotes to be able to replicate on their own and to produce new, daughter cells, prokaryotic daughter cells. And then we're also looking at how we're able to create our own body cells, okay, using mitosis to help produce genetically identical cells. Okay. At this point, we're going to be kind of shifting gears for essentially the remainder of this semester, and we're going to be looking at sexual reproduction. Okay. And so sexual reproduction is going to be where we're going to be able to create genetically unique cells. Okay. And so we're going to kind of introduce this a little bit today, and then we're going to look at it in a lot more detail on Thursday. So sexual reproduction is going to be involved with helping us to produce genetically unique cells. Okay, so before we get into that, another question here. So what does ploidy refer to? Okay, does it refer to the number of chromosomes in a cell, the number of sets of chromosomes in a cell, or the number of chromosomes that line up at the metaphase plate? And this is something we talked about on Thursday. Okay, what do we think? So Tim says B. Yeah, Christopher says B too. Yeah, so I'm seeing a lot of B's here. Perfect. So yeah, B is correct. So this is going to be referring to the number of sets of chromosomes. So remember, we were dealing with haploid cells that have one set of chromosomes and diploid cells that have two sets of chromosomes. So those are categories of ploidy. Okay, so ploidy is going to tell you the number of sets of chromosomes in a particular cell. Cool. Okay, so what we're going to be doing today is we're going to be looking at life cycles of different types of sexually reproducing organisms here. Okay, and so we're going to be looking at animals, we're going to be looking at plants, we're going to be looking at algae, and a lot of different other things as well. Okay, so before we get into all of that, there's a couple processes that are going to be important for sexual reproduction to help us produce offspring. Okay, so this is going to be a process that helps us to produce children, right? Children, offspring, babies, things like that, seeds, so on and so forth. Okay. So sexual reproduction is going to be involving two processes. Okay. We're going to be using fertilization, and then we're going to be using meiosis. Okay. So we're going to start by looking at meiosis here. Okay. And so in meiosis, what we're going to be doing is we're going to be looking at a process that looks very similar to mitosis. Okay. The difference here is that instead of having just one division and forming two daughter cells, we're going to have two divisions, okay, or two versions of cell division, and we're going to end up forming a total of 4 daughter cells here. Okay. Through the process of meiosis, what we're going to be doing is we're going to be taking a diploid cell, okay, and that diploid cell is going to eventually end up helping us to produce 4 haploid cells. Okay. 4 cells that have just one single cell. Okay. These haploid cells are going to be what helps us form gametes. Okay. So gametes are the sperm and the egg cell. Okay. So during meiosis, okay, meiosis is a process where we're actually going to be forming our haploid sperm or haploid egg cells. Okay. And so we're going to be essentially taking the chromosomes that we had and dividing them in half. Okay. So instead of having 2 sets of chromosomes, we have just 1 set of chromosomes. Okay. So that's the first process involved with sexual reproduction. Okay, the forming of the sperm and the egg through meiosis. The second phase, or the second process here, is going to be fertilization. Okay, so fertilization is where we take those haploid sperm cells or the haploid egg cells and we fuse them together to form a diploid zygote. Okay, so that's what we see here. Okay, so we see our sperm cells. Okay, the sperm cells are going to be haploid. This is going to be one gamete. Okay, the egg cell being the other gamete. Okay, they each contain 1 set of chromosomes. When they fuse, they're going to form a zygote that has 2 sets of chromosomes, 1 coming from each parent. Okay? And so we end up forming a diploid zygote from 2 haploid gametes here. Okay, this process makes sense. Any questions here? Okay, so meiosis and fertilization both have to be used together. So meiosis is going to help us produce the sperm in the egg cell. Okay. Depending on which sex you are, and then meiosis is where you actually take the sperm and fuse it with an egg cell during the process of sexual reproduction and intercourse. Cool. Okay. So when we're looking at different types of organisms, there's 3 different types of life cycles that we're going to be seeing when we're looking at multicellular organisms. So at this point, we're not looking at prokaryotes at this point. Okay, we're just looking at eukaryotes and different types of eukaryotes here. Okay. So there's 3 different types of life cycles. There's certain organisms that are going to be classified as diploid dominant life cycles, where they have a very short or no multicellular haploid stage. Okay. There's a second one called the haploid dominant, where all of the multicellular organism does not have a diploid stage. Okay. And then there's going to be alternation of generations, where both the haploid and diploid multicellular stages are going to be present. So we're going to take a look at what this exactly means for each of these individual life cycles. Okay. So the first ones are diploid dominant organisms. Okay. So diploid dominant organisms are usually going to be humans and then most animals here. Okay. What diploid dominant means is that when the organism has multicellular stages or multicellular components to it, those multicellular organisms are going to be mostly diploid. Okay? The majority of its lifespan is going to be spent as a diploid organism, okay, having 2 sets of chromosomes. Okay? And so with these organisms, with humans and animals, the only haploid cells that we're going to see are just the gametes, okay? The sperm and the egg here. Okay? And so what these life cycles typically look like is we're going to have, for example, our haploid sperm and our haploid egg. Okay, they're going to fuse, okay, through fertilization, and they're going to end up forming a diploid zygote. Okay, once they form that diploid zygote, they're going to start undergoing mitosis and start producing other diploid cells. Okay, so the majority of that human or that animal's lifespan is going to be spent as having 2 sets of chromosomes. The only time you're going to end up seeing a haploid cell is when that human or animal undergoes meiosis and produces the haploid sperm or haploid egg. Okay, so a very short period of time is having a haploid cell, okay, and the majority of its lifespan is going to be spent as a diploid organism. Cool. Any questions on diploid dominant organisms? Okay. So it's going to be most animals here, most animals and most humans. Okay. The second stage is what we refer to as the haploid dominant organisms. Okay. So this is going to be in mostly fungi and then algae as well. Okay. So these are going to be typically the only haploid dominant organisms we're going to see. Okay. So what's going to be happening is when we have a multicellular organism, the majority of the cells are going to be haploid, okay, only containing 1 set of chromosomes. And so what we'll see is we'll find 2 haploid cells, okay, that are going to end up fusing and then forming a zygote here. Okay, so here we have one haploid organism here, another haploid organism here, and they're going to end up joining together and they're going to form a diploid zygote, okay, the zygospore, okay, which is what we see in fungi. Once we form this zygospore, the zygote then immediately undergoes meiosis and then forms these haploid spores. Okay? Then from there, these haploid spores will then end up forming mitosis or going through mitosis, end up forming more of these little hyphae, which are almost like the roots for fungi. And then they go through this whole process all over again. Okay? So the majority of this lifespan is going to be spent as having haploid cells. Okay? It spends a very short amount of time as a diploid organism. Okay, that's haploid dominant. Okay, then the last stage is called alternation of generations. And so in alternation of generations, this is going to happen in mostly plants and some algae. Okay, and so what happens in alternation of generations is you essentially have organisms that spend approximately the same amount of time as having a haploid stage and a diploid stage during the course of its lifespan. Okay, and so it's going to be happening. We're going to see both haploid and diploid stages over the course of that lifespan. So what we're going to do is we're going to start here by looking at the gametophyte. Okay, and so this, like I said, this will be in plants and algae. And so the gametophyte is a haploid multicellular plant, and it's going to produce some gametes. It's going to be producing its own sperm cells and its own egg cells. Okay. Once they form that sperm and the egg cells, okay, they're going to undergo fertilization. Okay. they're going to undergo fertilization, end up forming a zygote. Okay. Now, it's not going to undergo meiosis because it's already haploid. Okay. You cannot split, one set of chromosomes into like half a set of chromosomes. Okay. They're already haploid, so it's not going to undergo meiosis at this point. Okay. So we're going to form our sperm and the egg, okay. And then the fertilization of the sperm and the egg helps to form the diploid zygote. Okay. From there, the zygote then undergoes mitosis, and you end up forming a multicellular diploid cell, okay, or diploid plants called the sporophyte. Okay. The sporophyte will then start to undergo meiosis. Okay. It has certain structures on it that are going to end up undergoing meiosis, and when you undergo meiosis, you form these haploid spores. Okay. And then those spores will eventually develop into gametophytes. Okay. So this entire life cycle is going to have about the same amount of time as haploid stages as it will in diploid stages. Okay. Okay. So those are alternation of generation. That is going to be found mainly in plants and then some algae as well. Okay. Any questions on these types of life cycles? Okay. So if not, we have a question here. So question. So which of the following best describes the life cycle of a rabbit? Okay. Is this diploid dominant, haploid dominant, or alternation of generations? Yeah, so I'm seeing A, so that would be exactly right, diploid dominant, right. So humans and most animals are going to be classified as diploid dominant organisms. Okay. They're going to spend the majority of their lifespan as multicellular diploid, containing multicellular diploid cells. Okay. And so the majority of its cells are going to be diploid, having 2 sets of chromosomes. The only time you have 1 set of chromosomes is when you're forming the egg and the sperm. Exactly, beautiful. Okay, next question. So we observe an unknown type of cell replicate. And we notice that the cell is diploid for a very short period of time and then undergoes meiosis. Okay. What type of life cycle best describes this cell? Is this diploid dominant, haploid dominant, or alternation of generations? All right, so I'm seeing B's, right, and that would be exactly right. It would be haploid dominant. All right. Okay. All right, and so that is that. Okay. So that's our entire lecture for today. Sorry, it was kind of all over the place. I apologize. I know we talked about 3 completely separate, different topics. But this chapter will kind of lead us into, or this kind of last section will lead us into meiosis, which we'll talk about on Thursday. Okay. So Thursday we'll talk about meiosis, this kind of major stage involving the life cycle, and then we're going to look at some different defects that can happen through the process of meiosis as well. Cool. Any questions on anything so far? Okay, so if there are no questions, there's some classwork that has been uploaded onto Canvas, so make sure that the classwork gets completed by midnight tonight. It's a pretty short, just like normal, so completed by midnight, you're good to go. If you have lab, I'll see you later. Yeah, that's pretty much it.